Hi, everyone. Welcome to episode 107 of Cell and Gene, the podcast. I'm your host, Erin Harris. Today's episode dives deep into a topic that's front and center for many gene therapy developers, how we're using large animal models, particularly non-human primates or NHPs, in preclinical testing. My guest is Ralf Schmid, Ph.D., Associate Director of Preclinical Research at Novartis. I think you'll glean from this episode that Ralf has a strong track record across the full drug and therapy development lifecycle from early stage discovery to clinical implementation. He specializes in the ideation and development of genetic and biologic therapeutics, with a particular focus on cutting edge genetic therapies for CNS disorders. Ralf and I talk about everything from the challenges of sourcing NHPs to the growing interest in alternatives such as pigs and sheep, to what the future might hold for preclinical testing in gene therapy. Plus, Ralf gives us a preview of the Next Generation Gene Therapy Vector Summit coming up later this month, and many more exciting innovations on the horizon. So stick around. This is a timely and thought-provoking conversation with Novartis's Ralf Schmid. Enjoy. Hi, Ralf. Welcome to Cell and Gene, the podcast. Thanks for being here.
Hi, Erin. Nice to be here. Thanks for inviting me.
Absolutely. So, okay, a couple of things. First, tell us a little bit about your professional background and the upcoming Next Generation Gene Therapy Summit conference coming up.
Yes, I'm right now Associate Director for Preclinical Research with Novartis Biomedical Research in Cambridge, Massachusetts, where I lead several of the gene therapy programs, assess the teams to really push those gene therapy programs, hopefully to the clinic soon. We have some exciting programs in the neuroscience space. Before I joined Novartis, for several years, I worked with Jim Wilson at the gene therapy program at the University of Pennsylvania, where Jim laid a lot of groundwork for gene therapies, where the gene therapies we're seeing coming towards the clinic right now, and I really learned basics there, which I'm trying to apply now in industry. And thanks for asking about the conference. So, it's an important part still. I think it should be an important part of every researcher's life to go to conferences and exchange ideas and hear new ideas. So I will be attending and participating in a panel discussion at the fifth Next Generation Gene Therapy Vector Summit, put together by Hanson Wade in end of July, so that's July 29th through 31st in Boston. It's a great conference to learn about cutting edge new gene therapy vectors, how key stakeholders in the industry think about moving programs into the clinic, safety considerations, biomarkers, and things like that. If you want to find out more, the website is very easy. It's nextgengenetherapyvectors.com, and you can find out more information there.
Wonderful. Thank you so much. And yes, listeners, be sure to visit that website to learn more about the conference. We'll be talking about it a little bit here and there throughout our conversation, but definitely check that one out. It's going to be a great event. So Ralf, talk to us a little bit about what recent news or innovations in gene therapy that you're most excited about.
Well, I think most of us have been really excited about the next gen AAV capsids being developed by several small companies, but also by academia. And so recently there have been several announcements that companies are either finishing up their preclinical package or even have an open IND. And so I'm really excited to see some of those companies entering the clinic, because the expectation really is that this is an absolute game changer for neuroscience in terms of biodistribution of the gene therapy payload in the brain.
Good. Okay. Going back to our conference that we talked about a little bit, how will your role and the role of the 25 other speakers at the conference enable these innovations that we're referring to perhaps progress even further?
Well, we had actually not that long ago a special, more specialized conference, and I think that will be put on again in the fall, looking at those next generation capsids, at the different ones which are out there. So what this conference is more focusing on, okay, now you have something really cool, you have something which has great clinical promise, now what? So how do you really implement that? So how would you, what are your best options? What 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 are the opportunities for small animal study, large animal studies? What you need to file your IND? What are your safety considerations? What are your regulatory considerations? It's really, I think this conference will help people to better understand what the framework is, even if you're already working on this. So you might learn something new, make some new connections with other attendees, to hopefully drive your work forward.
Great. Okay. We talk a lot about non-human primates on Cell and Gene, and so I would love to get your perspective on that particular topic. And so my question for you are, what are the biggest scientific or logistical hurdles you've encountered when relying on non-human primates, or of course NHPs, for preclinical safety and efficacy work in gene therapy vectors?
Yeah, first of all, non-human primates have a very dominating role as a large animal models, and I think most of the genetic medicine modalities, including gene therapy, um, because the size and the evolutionary distance is closest to the humans. So the thinking is still, that this is best to study both the biodistribution or the expected efficacy of your gene therapy, but also safety aspects. And I think the field has kind of narrowed in on those models, and there's a lot of specialized CROs out there which can help you with the work. But now on the flip side, that has really increased the demand for NHPs dramatically. I mean, like 10 years ago, there was very little demand, and then it suddenly increased a lot. And since the pandemic, it has become a huge problem to source enough of those NHPs, because the sourcing environment has dramatically changed since the pandemic, where several countries are not allowing exports anymore in Asia, where they normally came from. And demand has increased at the same time, so that had negative effects on price. And also from the more practical research side is that you never know what you get. So if you want to do your studies, you have to, we're relying on CROs, and they're trying to source as best as they can. But what we had to find, it makes a difference from which part of the world those NHPs come from, in terms of both size and physical health, but also how they react to gene therapy. And that's a little bit unfortunate that studies are a lot more complicated these days with NHPs.
Fair. Okay. Talk to us a little bit about the timeline impacts of NHP studies compared with alternative large animal models. What does that timeline look like? Excuse me. And then how do those pressures influence project, do we do it, do we not, type of thing, decisions inside big pharma?
Yeah, so NHPs, they typically come into the game fairly late. So after you have identified leads, after you're pretty sure, that your gene therapy is going to work out. So that's kind of the last test, really. And typically you will need to do a pilot study to really determine dose and, look at early signs of toxicity before engaging in a much larger and much more expensive GLP safety tox study, which is a requirement for filing an IND in most cases. So Practically, it is now that sourcing of the animals is very difficult, and so you have to make reservations far ahead of time, especially for larger studies. So, if you want 20 or more animals from the same colony, from certain genders, and so on, you sometimes have to plan ahead 6 to 9 months before you want to do your study. You have to place an order with a CRO to give them enough time. NHP breed only once a year, so it's a complicated animal to work with because it's not like mice where you can generate a lot of them in a very short time, and so that now is complicating really planning for everybody. Because sometimes you're not actually sure. I mean, when do I want to do my first NHP study? So am I going to be ready in 6 months, in 12 months, or in 18 months from now? So if you log into a slot and then, anything doesn't check out, most of us cannot spend, something in the order of $500,000 or $1 million in testing a somewhat lackluster lead. So you want to test your best possible lead. And so I think it's dramatically affecting the turnaround time to really get, move forwards towards the IND. It's not only the cost, but it's this whole time constraints.
Sure, of course. And I want to talk a little bit about the ethical side of this. So we know that ethical scrutiny of primate research keeps intensifying. And so how is the industry weighing replace, reduce, refine, or the 3 Rs in this space? And where do you see the most immediate opportunities to replace primates altogether?
Yeah, that's a good question, and thanks for that concern. I completely support that, and unfortunately, there are some bad players in the industry, especially on the sourcing side, where animals are literally abducted from their natural habitat and then falsely labeled and sold on the US market. So that I think that's still occasionally happening, and that's absolutely not okay. But that's of course, influencing also the kind of industry aspect where we really try to, both from the breeding, transport, and usage to think this through and reduce the use of NHP as much as we can. So I would say, everybody is really considerate and should be considerate of how many animals you use, how you design your studies, that you design your studies that they're really meaningful. So that is not something, you say, oh, let's just do another NHP study, but it's one study. You carefully design what kind of doses you use, what kind of time courses you use, collecting rather over collecting tissues and bodily fluids that you can get as much as possible out of those studies, and I think also be courageous and say, well, we're not ready. We rather give the monkeys back and let somebody else use them. So affecting overall use. Unfortunately, I don't see right now that we can fully disengage from using NHP. But one is, of course, the safety aspect, because that would just mean, you're not using NHPs, you have to use another large animal. That's another discussion, really, using a sheep versus a pig versus an NHP. So if what the ethical considerations or preferences might be here. But I think, this whole thinking that we can do everything lab on a chip or, in a dish or anything like that, it will be difficult because we are not very good in predicting this last mile of safety events. Of course, if it's drastic safety issues, you can pick that up early. You can pick that up early on. But I mean, for example, immune reaction, a response of the immune system, that's very difficult to model, and I think we really need that insight for the safety reasons when going into patients. And then a lot of our therapies these days, in the AAV field, they rely on, at least the newer therapies rely on AAV capsids which cross the blood-brain barrier. And so that is also very difficult to model. And it's also, even if you can model it, and people have made progress here, it would be very difficult to predict what your actual biodistribution is in the brain. And a gene therapy is a one-time therapy. Right now, we don't have the tools for redosing, so we have to get this right. And so the only way I see right now is really, do one meaningful experiment in NHP, show we got the right dose, we have the perfect biodistribution. Now we can move. We can move in the patient and would expect that it has a significant therapeutic effect on the patient.
Sure. Okay. Earlier you mentioned certainly pigs versus sheep. And speaking of pigs, they often mirror human anatomy and physiology. Which specific safety and pharmacology endpoints have you found pig models to excel at? And where also do they fall short versus NHP?
Yeah, I have personally not used pigs in my research, but I know in my field several researchers rely on it. I think pigs are a really strong choice if you think you need a actually genetic model. What I probably should have mentioned, all the NHP are being used, or I've been referring to, are wild-type NHPs. So they are not a disease model. So you can only study the more biophysical parameters and of course safety of your therapy. So if you're actually interested in, seeing in a therapeutic response and using as kind of as a mini patient, then pigs would be a good choice, because these days we have the tools to genetically modify pigs to make them an appropriate model. Disease model. Of course, it's another question if you want to do that, because a lot of the diseases we're working on have very severe symptoms, which would drastically impact the quality of life of the pig. So that's another ethical consideration. But just using wild type pigs, there are some benefits that, as you said, both the immune system, metabolism, and other aspects are closer to human actually, or close enough to human that pigs have been used a lot for small molecule research and other modalities. So we have a very good understanding how results translate. The experience with AAV gene therapy is not that big right now with pigs, and we don't understand, for example, if the blood-brain barrier of the pig, how that would resemble or not resemble the human blood-brain barrier, how biodistribution in the brain would translate into humans. So I think if the industry wanted to use pig as an alternative, wild-type pig as an alternative, I think more work has to be done to really fully establish that, and also doing comparative studies between pigs and monkeys to kind of understand how they relate to each other. But I think it's a good idea, and of course, depending on the part of the world you're from, there's others, especially in Australia and New Zealand, who heavily advertise sheep as a model. So that's another idea, of course.
Sure. What best practices have emerged from generating pig data that regulators deem perhaps credible and translatable, whether that's dosing strategies, biomarkers, and imaging endpoints? What would you say are those best practices?
Well, I think okay. So I think any of those biomarkers or imaging studies. So I think they're not specific to pigs, but they would apply to any large animals. Also, to if you had a dog or cat models. So sometimes those are available. So and I think it's on a case by case basis. So it's not that regulators say do a study with a specific animal and we wave you through. So it really depends on justifying why you want to use a specific animal. So you could say, I think, for reasons X, Y, Z, pigs are much better models for my particular disease or my particular approach than NHPs, and that's why I chose to use them for my IND enabling studies, or any of the other models. But it also boils down to expertise because depending on what you want to do, how some gene therapies rely on other routes of administration than IV dosing. So if you're thinking about intrathecal or even direct brain injections, so you also need to have the expertise, the veterinary expertise, really to use those models. And I think those are probably the bigger considerations that you're able to find somebody who can do this under GLP guidelines.
Sure, sure. Okay, just one final question before we head out here. Looking ahead, perhaps 5 to 10 years, what do you think the default large animal model will be for safety and efficacy testing? Do you see pigs becoming the standard? Do you think it will be a mix of models? What's your take?
That's an interesting question, and of course, as we discussed before, ideally we would have not any large animal models became the standard here, but find a smarter way to do this. But as I also said, probably we have to, for the next decade, we have to rely on those large animals, and so I think. As I mentioned before, it would be a good idea to start diversifying in the use of animals. It might also help you to better understand your safety signals, your biodistribution, if you're using more than one animal, because we just simply don't know. I mean, just because most of the INDs are based on NHPs, that doesn't mean that this is the best choice. So it could be it's either another monkey species is a better model, is also better to source, might be more affordable, or it could be that establishing either pig, sheep, anything along those lines. And I think pig is also like what kind of pig are you exactly talking about? Because there's also different species. So I think people need to work through that and really identify what are the good alternatives, why are they, and then pushing hard to include them in studies. What I'm a little bit more concerned about is kind of the recent push by the NIH to get rid of any kind of animal research. That might work fine for certain areas, but clearly not for one-time therapy, which we cannot switch off, which we cannot get back out of the patient, or even thinking ahead if it's any kind of gene editing modality where directly the DNA in cells in patients would be altered. So I do think that we need appropriate large animal models where those approaches can be fully tested, that we fully understand this before trying out in a patient.
All right. Thank you so much, Ralf. That wraps up this episode of Cell and Gene, the podcast featuring Ralf Schmid, Ph.D., Associate Director, Preclinical Research at Novartis AG. Ralf, thank you so much for your time and your expertise. It was wonderful having you. And I'm sure we want to tell our listeners to look up and register for the 5th annual Next Generation Gene Therapy Vectors Summit coming up next month.
Yes, thanks, Erin. It was a pleasure talking to you, and I hope that I will see some of the listeners in person at the Next Generation Gene Therapy Vector Summit at the end of July in Boston.
Wonderful. All right, listeners, thanks again. Visit us at cellandgene.com, and we will talk to you soon.